If you have any questions, there will be a Q&A at the end, as well as my email is at the bottom here, support@nexcelom.com. You can always reach out to me afterwards. So getting started, welcome. Okay. We're going to go over briefly the Cellometer Vision CBA, and we're going to get familiar with the software. We will create a new assay and edit an existing assay. We'll go over how to create a new cell type and how to edit an existing cell type. I'll also go over user times, and I'll do a live demonstration to summarize everything we've learned so you can see it. So, the Cellometer Vision CBA has user exchangeable dual fluorescent channels. It's suitable for primary and cell line analysis, and we recommend using AOPI for viability measurements. So Tricam Blue is available for cell lines and purified primary cells. Other assays it can do is a GSP-RC transfection efficiency assay, a GSP-PI viability assay, cell cycle PI, and autophagy, which was the highlight of our month. So these user exchangeable FOMs, or fluorescent optics modules, range from emission filter 470 to 695. 2 optics modules can be installed at any given time. And here are just a couple examples of what each filter can detect. So this assay that we're going to go over later will be a 535-660 assay, which is usually what our instruments come with. That's a green and a red channel. So just so you guys know, these are just a small little fraction of an example of the cells that we can count with the vision: PBMCs, splenocytes, stem cells, tumor cells, and very many more primary cells, as well as different cell lines, such as JERCAT, HEC293, HeLa cells, and so many more. And just in case you guys ever have a cell type that we do not have in our library, you can always call in and we can optimize cell type for you. So getting more familiar with the software itself, here's our home screen. This should be familiar to all you current users. We're just going to go through all the little different buttons and areas, beginning with our assay setup. So here we see your chosen assay. This is your drop-down menu. And this little pencil is what you'll use to edit the assay. Below that, you have the cell type that is associated with your assay. One second.
Just my screen.
Okay, sorry. The cell type that's associated with the assay and how to edit it, and the imaging mode that is also associated with the assay. So in this case, we have an AOPI assay that uses an F1, F2 imaging mode. So next is the sample ID and dilution factor, where this is where you set your ID and dilution factor, which is very important for data managing. You have your preview bright field button and your preview F1 and F2 fields. And just below there, there's also the exposure time. On to the right side of the screen, this is where you can view your image. We have 4 images to every sample, A, B, C, and D, which can also further be looked into with the 1, 2, 3, 4 quadrants. And you can also zoom in on these quadrants if you really want to get very in-depth into your analysis. Below these views, you can toggle between the bright field 1 and bright field 2 screens, as well as your fluorescent 1 and fluorescent 2 screens. Once you're looking at your cells, you can combine your F1 and F2 images. For example, if your AOPI can look at what's live and what's dead at the same time. And you can also find the outlines of what's counted and what's not with this counted box checked off. Moving to the top of your screen, this first button will bring you to your results. So you immediately get your results, you X out of it to look at your images, and this is how you get back to it. The second one is your sample calculator. It's very effective if you're trying to get a target concentration or if you're looking for a certain number of cells to place. And third, we have your cell size histogram, which will show you the diameters of all your cells counted and a size distribution histogram. And here's your results template. So again, I hope you guys are familiar with it. we have up in the top left corner the assay and cell types that were used. We have the time and date of your sample that you ran, and the ID and dilution factor of that particular sample. Move down to the results. In this case, we only have the F1 and F2, but in an updated software, you will have your total cell count as well. We have the count, concentration, and mean diameter. And we also have the viability right below that. And if you're not using FCS Express to analyze fluorescence, we do have mean intensities below your viability. So we're going to move on to creating a new assay, and how it's kind of the same as editing a new assay. So, step one, you're going to select the initial assay type in the assay drop-down menu. And then you want to click on that pencil icon next to the assay menu I showed you. And the first thing you want to do is save as a new assay type. And here's where you'll name it for whatever you're doing. In this situation, we're going to develop an AOPI assay for PBMCs. So we've put in PBMC AOPI viability assay. And then you want to select your imaging mode. And for AOPI in this case, it's going to be fluorescent 1 and fluorescent 2. The other imaging modes available to you are the bright field only, which uses only bright field to count cells and measure size. The fluorescent only, which uses only the fluorescence to count cells. There's a bright field and fluorescence option that uses a combination of both images to count your cells. The dual fluorescence image mode is for assays where double positives are likely, and we want that to happen. These are not for viability assays. It's more for like GFP, RFP cells. And there's also an option of multi-mode counting, where you can measure the fluorescent intensity only within the cells located in a bright field image. So once you've chosen your imaging mode, you're going to choose your cell type for both the F1 and the F2 image. So we have the entire cell library in these drop-down menus. In this situation, we're going to choose our PBMC fresh samples. And now that you have selected your F1 and F2 channels, we can further develop your exposure times. So here we have 500 and 4,000, which are the default, but it's important to know that each cell sample is different. So while we can start here, it may definitely take a couple, goes to get the correct exposure time for your sample. So after all that, we're going to save, and we're going to choose that assay, and just do like a test run. So this, for example, is an image of an AOPI assay with the combined checked off. We see red cells and green cells. And this is how you'll know if you need to adjust parameters or exposure times. So we're going to now move on to the cell types. So the cell type wizard is a tool in our software that uses either a saved image or a live image of your cells to analyze and create parameters for accurately counting your cells. The user has the ability to name the resulting parameters and save them for future counts. So it's not a one-shot go. You can save this and keep using it. You could also save a new cell type from an existing cell type. So just like in our assays, we have a little box save as new cell type and rename it. If you're ever playing around with cell types, it's always important to save as a new cell type before anything gets lost or mixed up, because sometimes it's very difficult to go back and know exactly what all your different parameters said. And again, if you ever do want to optimize a cell type, it's no problem if you just call in and we can work together, or you could email in images as well, and I can do this for you. But if you're in the moment, I get you want to make some adjustments. So we're going to talk about some of the parameters. Please call in. I'm just saying, always save as a new cell type. And I'm going to give you a brief overview of these parameters. So please, call us. It'll be okay. It's what I do, and I love to do it for you guys. So starting with the cell diameter, And this is for the bright field and fluorescence. It depends on your assay. For example, our AOPI assay is for fluorescence 1, fluorescence 2, and you'll need to make these in the fluorescent tab, which we'll talk about later. But for now, in the bright field, you can set your min and max diameters, and this can help you gate out cells or distinguish different cell populations. For example, SF9 cells, the bigger cells producing protein can be distinguished from the smaller cells not producing protein. Next is the roundness. All shapes of cells are included at low values. So if you set your roundness at a low value, all different shapes of cells, not just your round ones, will be included. Roundness is a tricky one. I would not recommend changing this often or making them perfectly round. And you can see our default here is 0.10, and our range is 0 to 1. So the default is usually pretty safe to go with. Next is the contrast enhancement. When you have a low value, it means the cells have a high contrast to the background. A high value will count low contrast cells. And all these parameters, this is just a very brief overview, and there will be, in the future, a webinar to go very in-depth into these features. And if you have any questions on it, you can definitely contact me at the end of this or at a later time. Moving on to the declustering parameters, there's an option to not decluster clumps. Only check this if you do not want to count individual cells within a clump. The clump will be counted as one big cell if it falls within the cell size parameters. Next is the decluster edge factor, which indicates the degree in which the software needs to enhance the cell edge in order for the declustering to happen. Use a high value when cells don't have well-defined edges. The declustering pH factor is the threshold ratio between the cell signal and the background. A high value indicates the cell signal to the background is high. Background adjustment is the threshold adjustment needed to pick up strong or weak cells, and a high value will indicate the cells are well-defined. Moving over to the fluorescence parameters. Again, you see the cell sizes here and the roundness. So if you're counting in fluorescence only, these are where you want to make your cell size changes to do your gating or whatever you want to do. But for the other parameters, we have normalizing intensity for cell size, which is, as it says, fluorescent intensity is being normalized to the size of the cell. Non-uniform cell fluorescence. If a portion of the cell has a fluorescent intensity above the threshold, then the cell is considered positive for fluorescence. And this is useful if you're doing punctile staining, for example. There is an advanced VRF mode, which helps to improve the declustering within the fluorescent channel by using the bright field, almost as a mask, in case you have some very fluorescent cells. The threshold is when the software finds the brightest fluorescent spot on the image and attributes the value of 100% intensity to that one cell. And the software will then exclude any cells within an intensity measured below that given threshold percent value. So if you set your threshold at 5, any cells that are less than 5% as bright as that first cell will not be counted. So now I'm just going to speak very briefly on the exposure time. So if your cells are dim, you need to adjust your exposure time. If your cells are too bright, adjust your exposure time. This has like a really big effect on your counting. And here I have an example of 2 dim, just right, and 2 bright cells. The first image is very dark. It's hard to make out your cells. They're kind of diffuse looking, and the background is too dark. Your 2 bright cells on the far right side. You can see the background is kind of too bright. It's a little blinding. And your cells are also kind of changing their morphology. The fluorescence is kind of moving out of them. So in the middle, we have a nice clear background. It's not too bright, not too dark. And you can clearly see your cells. So if you ever need to adjust these exposure times, return to the assay settings and to the F1 image and the F2 image to reset our cell type and adjust accordingly. So like I said, each cell type is going to be different, and these are just basic starter-off parameters, but feel free to play around with them. So now I'm going to do a live demonstration. Software. Okay, this is a preloaded image of PBMCs, actually. So I'm just going to recount them. Okay, and we have our results table, and we see our AOPI assay, our cell types, my sample ID, and dilution factor. We just did it, and I have a viability of 87.9, pretty good. Our concentrations are pretty good. The concentration you want to keep your cells at is around 2 million, 2 times 10 to the 6. So also from here, we can go to your sample adjustment. We can save from here, and we can show the sizes. We can export, print. Moving back to the main field, we can go back to results here, just like that. Open our sample calculator. If you've never seen it before, we can go to our histogram for size. I have a lot of cells. And here are all your diameters for each cell counted in your histogram data. We can go through the different ABC images. We can get a closer look, zoom in further. We can see what was counted. And if you're ever in a situation like this where these are nice bright cells and it's hard to see the green outline against the green background, you can take the tint off, and now we can clearly see which is outlined in green. Just an FYI for you guys. Look at our bright field 2, our fluorescent field 2. See what's counted. We can see combined. So let's go into our assay now. When we see we have our AOPI assay, fluorescent mode, we acquire a bright field. Our cell types are here, and we have our 535 filter set and our 660 filter set, 500 and 3,000 exposure times. Unfortunately, because this is a saved image, I can't do any bright field adjustments or exposure adjustments for you guys to see the difference. But we can also, just for fun, make a new assay type. So this is your blank assay template. So we want to save as new. And we can do, for example, AOPI test. If there's a further description needed, go ahead and put it in. So this is for our webinar. So we want to change our imaging mode to our F1, F2. And now we have these options for our cell types. So I'll just pick a jerkat. I like jerkat. Well, and now we want to specify our fluorophores, our filters. So 535 is our green channel, and 660 is our red channel. And our default exposures are around 500 and 4,000. But again, these should be adjusted and played with. And you are using modules. This wouldn't change unless you're doing some high sensitivity work with, for example, surface markers. Something else the Cellometer can do. So we can save this. Now we have our new assay type, and we can test this out. And here we go. Counting results right there for you. But let's see what was counted. So maybe we toggle between, this is a PBMC sample, not a jerkat sample. So our parameters are going to be off, right? So we're going to make adjustments to these parameters, just like we learned. So we take a good look. Maybe, I don't know, I want to count another small cell. Go into here. We can edit your F1. And we're going to use the fluorescence because that's what we're counting. So we can drop down our size. We can increase our size as well. And put it up to, I don't know, 30. Our threshold looks good because nothing got left out pretty much, right? We want to make the same adjustments at the same time to the F2 field. And that's why we can go back and not forget what we put in. It's the same, 3 to 30. Okay. So we can recount and see how that affects our counting. Okay, so take our tin top. I'm going to check my parameters again. I'm going to take off multi-mode because I don't want to count in bright field, which it was doing. So just changing the size around is one of the things you can do. We can exclude a bunch of little cells. We can bring it up to 10, things like that. So this is just a very brief introduction on how to adjust your assay types and cell types. I hope it was informative for you. If you have any questions, please contact me. Do not hesitate. We'll check this really quick, and then we'll open it up for questions. Just look at all those ones that got excluded. Whoa. Okay, so learned our live demo. Any questions?
Great. Well, thank you very much, Stephanie. That was a wonderful presentation. So as we did mention, if you have any questions, go ahead and type them into your questions bar, and we will get through them one at a time. We do have a question. It's a bit lengthy, so I might say something wrong, so if needed, I can repeat it. So our first question is, in the 2 examples you just did, you used 2 different types of formulas, F1, F2, F1 times 100 versus, or F1 minus F2 divided by F1 times 100.
I'm going to write this down.
I can show you here too. So it was F1, F2, F1 times 100 versus F1, F1 plus F2 times 100.
Gotcha.
Is there a setting, in the setting, there's also F1, F2 times 100. And this is a bit confusing. Which one should we be using?
Well,
so Raid, thank you for your email, your question.
Thank you for your question. There are a bunch of formulas depending on what you want to do your assay for. The correct equation for viability assays, AOPI specifically, would be F1. Hold on a second. I need to see it.
So the one for viability is F1 minus or F1 divided?
F1 divided.
F1 plus F2.
F1 plus F2 divided by
F1.
F1.
Times 100. Order of operations.
Order of operations.
So that's for viability. So where would we use F1 minus F2 divided by F1 times 100?
So to answer this question, I'll field this one. This is Dimitri. I'm staff scientist here. So a lot of these formulations really depend on this particular assay that you use and the fluorescent optics modules that are installed in your cellometer. This particular instrument, we did a 535-660, which means there's absolutely no crosstalk between the 2 filters, and the 2 populations that you acquire are the green populations, which is live cells, and the red populations, which is dead cells. To obtain the total cell count, we need to add those 2 populations, dividing by the live cells to give you the % viability. When you have a different filter set, such as 535, 595, there is a spillover or crosstalk between the 2 signals. So we use the F1 as a total cell count, and we subtract F2, which would be double counted in both the green and the red channel. We then divide by that number in order to obtain the viability. So the formulations don't really you have to be careful with which formulations you use depending on the assay as well as depending on the filter set. My suggestion would be to contact customer support at Nexcelom to answer any of those further questions.
Fantastic. All right. You are very welcome, right? So we have another question. This one's from Cam, and Cam is asking, is it critical to have an exact range of cell size? For example, dendritic cells are much larger than lymphocytes.
I'll fill this one as well. So it depends on the experiments you actually are conducting, whether you're looking at immature or, for example, or mature dendritic cells. So depending on the maturity level of dendritic cells, you will have an increase in cell size, which, as you stated, much larger. The key is if you're having a heterogeneous population that you're looking at and you're looking to exclude some of the smaller cell counts, then yes, you will have to have a tighter range to hone in on the larger dendritic cells. However, if you're having a more homogeneous population, then the cell ranges aren't as important. The cell size ranges are not as important. So Stephanie had mentioned earlier in the presentation the SF9s, for example. If you're looking for mature and immature SF9 cells, those that are producing proteins, those that are not, you're going to have a very clear cell size difference. So the tighter range you can set on smaller SF9 cells, or vice versa, you can choose to detect larger SF9 cells. So depending on your assay and the population which you're examining will directly correlate to the cell sizes that you choose.
Great. So I would answer that by not necessarily critical, but again, that's where adjusting our size parameters can help to make sure that we're including, when there's a big difference between two types of cells, we can adjust to make sure we're capturing all the ones that we want to get while still excluding, but it's not critical down to like the tiniest micron.
Yes, the key is we cannot, for example, differentiate different white blood cells. The cell sizes are too close, they're too similar. If you have a significant cell size difference between two populations, that's when we can distinguish the differences. But Sarah is correct. So most times we do not have to have a very tight range in our cells.
Great. We have another question from Debbie, and Debbie is asking, how do you diminish halos around the cells?
Fluorescent halos? Well, you can reduce your exposure time, for one. If you see a halo blooming, that's a clear sign that your exposure is too bright.
All right, so reduce your exposure.
And also your focus is a very important part of it. You're looking for clear defined membranes and bright white centers.
So Stephanie answered that question very well, but I would like to add on it something more. So if you're using a two-color assay, you must keep in mind that different fluorophores are excited in different planes. So a green and a red fluorophore would be slightly in different planes. So if you're doing a two-color assay and you're focusing in bright field, there's going to be a natural offset, and you may see a slight halo effect. So one of the ways you can look at it is click Preview, and simply preview your image in the fluorescent channel to assure yourself that you don't have perhaps a halo, and adjust your focusing accordingly. However, if you're doing a one-channel stain, a single-color stain, and you are focusing in bright field, then you should have much fewer or much smaller halo effect in a fluorescent image. Typically, the halos, as I said, are in a two-color. For example, if your green is 100% in focus, the red might be slightly out of focus. So you may have to tweak and adjust those parameters. As well as Stephanie said, please do check your excitation.
Great. So we have another question. This is from Reid, and it is, does the amount of time the cells stay in AOPI before the analysis affect signal intensity?
No, it's pretty much an instantaneous stain. What will affect intensity is if it stays in the slide. But that's a webinar for another time.
We have done multiple studies over a long period of time looking at the fluorescent intensity of 5, 10, 20, 30 minutes, and so on. And the stain in the dye is very stable. So incubating your sample in AOPI, although not recommended for extremely long periods of time, it is not as toxic as trypan blue, and the staining efficiency should not change after 5 minutes.
All right. Do we have any other questions today? I'm not seeing any other questions. If there aren't any, we can go ahead and wrap this webinar up for this month. As always, if you do have any questions that come up after the fact, please feel free to reach out to support@nexcelom.com. As I did mention at the beginning of the webinar, this has been recorded, so this will be available on our website through the same link that you went to log in today. Shortly after, we will send out an email with that link as well, so if you found this content interesting or helpful, informative, you can pass it along to anybody else in your lab. They can always register after the fact to access the recording information. And next month, we are going to bring you another wonderful webinar. Topic is to be determined. So please feel free, if you do have anything that you would specifically like to see training on, to reach out to us. We'd be happy to take that into consideration as we continue to bring these to you every month. With that, I will say goodbye. Thank you for joining, and have a great rest of the day and a great weekend, everybody.